So with Chapter 4, we had looked at cellular organelles. With Chapter 5, we're going to get in more in depth on the plasma membrane and the way that things are transported across the plasma membrane. So we've talked about all of our major macromolecules, our lipids, and our phospholipids are one of the main lipids in our body. Phospholipids are what make up our plasma membrane. If you remember, we talked about phospholipids with the polar head and the nonpolar tails, causing the phospholipid bilayer with the polar heads aiming out towards the extracellular fluid and also aiming in towards the cytosol. Along with the phospholipids in the plasma membrane, we have cholesterol that helps to stabilize the plasma membrane. We have proteins that act as receptors and act as enzymes, act as tunnels and channels through the plasma membrane. We will look in more detail all the roles that proteins play in the plasma membrane. We'll also look at receptors such as our glycoproteins and our glycolipids that help in cell-to-cell recognition. Outside and hooked to a lot of these proteins is our extracellular matrix. And on the inside, a lot of these proteins are hooked to our cytoskeleton, and in particular, microfilaments of the cytoskeleton. So as just mentioned, our phospholipids have a hydrophilic head, which means they love water, they are attracted to water. And there's hydrophobic water-fearing tails. And so they point towards the inside. This makes our phospholipids amphithetic. So those phospholipids are amphipathic, which means that they have a hydrophobic area and a hydrophilic area. Again, that is our hydrophilic water-loving heads, which have a phosphorus, and the hydrophobic fatty acid tails that are water-fearing. And because of that, they orient tails hidden in the center in this phospholipid bilayer. Plasma membranes are very dynamic. They are constantly changing due to the demands of the cells or the needs of the cell. And here is an example of an experiment that was run where we put a mouse cell and a human cell in close proximity. The two cells will eventually merge and mix the proteins that were on their cellular membrane surfaces. So a lot of those proteins can move horizontally in between and move around. With those phospholipids, if you recall back, you can have saturated, which mean that they are full of hydrogens. That gives you straight tails. They can pack closely together. But you can also have unsaturated fatty acid tails that give some kinking so that the phospholipids can't stay close together. And this is how plasma membranes vary throughout the eukaryotic domains. And even in the prokaryotic cells, the plasma membranes will vary. So if you were in a very cold environment, you would want the kinked tails that would allow the membrane to stay fluid at lower temperatures. So fish who are in the Arctic, their plasma membranes will have a lot of these kinked tails to allow for enhanced membrane fluidity in those colder environments. But when you have something like our archaea or bacteria that live in very harsh environments like the hot springs in Yellowstone, they would have very closely packed phospholipid membranes, increasing the viscosity of the membrane when they live in those 90+ degree temperatures so that their membranes would not lose structure in those high temperatures. In animal cells, we actually have cholesterol embedded to help give some stability and some fluidity to the phospholipid bilayer. And so at lower temperatures, it hinders solidification by disrupting the regular packing of the phospholipids, kind of like the kinking did back up here in our unsaturated fatty acids. So the cellular proteins that exist in the plasma membrane can have a large variety of functions. They can transport, so they can act in the channel or carrier proteins, so membrane proteins that transport ions or molecules across the membrane. Enzymatic activity. Signal transduction, so they can be ligand receptors where they will relay a signal from something binding on the extracellular side into the intracellular side. The glycoproteins and glycolipids that we talked about earlier also act in cell-to-cell recognition so that cells can recognize who's who and if something's self or if something's not self. Intercellular joining, so forming attachments in tight junctions or in desmosomes. Also our gap junctions, but those are more of a tunnel like our channel proteins. And then attachment to the cytoskeleton and to the extracellular matrix, as I pointed out in the first slide. So it allows the cell to recognize and connect to the outside to the inside. So allowing a continuous connection between the external environment into the internal environment. So carbs and their functions, that's what the glyco mean. Glyco means carbohydrate. And those cell-to-cell recognition proteins can be bound to proteins. That's where you get a carbohydrate bound to a protein. Or it can be glycolipids, where it's a carbohydrate bound to a lipid. And so here you can see a glycolipid bound directly to a phospholipid tail there. Our membrane proteins can be two main types. We can have integral proteins, which go all the way through the plasma membrane from one side to the other. So the majority of these are transmembrane proteins. And you can also have peripheral proteins, which are just surface proteins, and they tend to be associated with our integral proteins, but they are either on the internal side or the external side, but they tend to be more towards the internal intracellular side. The synthesis of our membrane components done through the endomembrane system. Remember back in chapter 4 we talked about the endomembrane system. So things that are processed or manufactured in our ER, proteins or carbohydrates, so things processed in either area of our endoplasmic reticulum, are transported to our Golgi, and in the Golgi they are modified. So the glycoproteins undergo further carbohydrate modification, so carbohydrates added on back here in the ER, but then further modified in our Golgi. And then the glycoproteins, glycolipids, secretory proteins, all transported in vesicles to the plasma membrane and incorporated into the plasma membrane. As I pointed out earlier, our plasma membrane is a very dynamic membrane or organelle of the cell, and it is constantly changing depending on the demands of the cells or the demands of the body at that time. A lot of the items that are also in these vesicles will also be excreted via exocytosis from the cell.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So items that move with their concentration gradient are going to move via diffusion. If they can move directly across the plasma membrane, It is simple diffusion, so they're going to move from an area of higher concentration to an area of lower concentration until they reach equilibrium. And you can have items traveling in both directions across the membrane, but they're always going to be traveling from an area of higher to lower, until they reach equilibrium.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is cotransport. Here, both sodium ions and glucose move into the cell through a cotransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
So lots of ways in which we move things across the plasma membrane. Passive diffusion, again, is moving with our concentration gradient. Passive diffusion of solutes are going to move from an area of higher concentration of solute to an area of lower concentration of solute. But when water molecules are moving because the solutes can't move, that is osmosis. And so water is going to move from an area of higher concentration of water to an area of lower concentration of water. And a lot of this is dependent on the solute to water ratio.
The plasma membrane is permeable to water molecules and allows the movement of water into and out of the cells, which is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. If a membrane is permeable to water, what happens when a solute cannot pass through the membrane? Let's experiment by placing glucose molecules inside the cell, outside the cell, or both. We'll try different concentrations of glucose on each side of the plasma membrane to see which way water will diffuse and what will happen to the cell. When a cell is in a solution that's more concentrated than its cytoplasm, it's said to be in a hypertonic solution. The solute molecules attract water molecules so that fewer water molecules are free to diffuse across the membrane. In a hypertonic solution, the concentration of free water molecules is higher on the inside of the cell than the outside. Osmosis occurs as water molecules move down their concentration gradient, leaving the cell. As a result, the cell shrinks. Here's another scenario. The glucose molecules have been distributed evenly inside and outside the cell. The cell is in an isotonic solution, where the concentration of the external solution and the cytoplasm are the same. The solute molecules attract water molecules. Since the concentration of solute molecules is equal on both sides of the membrane, the concentration of free water molecules is also the same on both sides. Water flows back and forth across the membrane in equal amounts, and so the cell neither shrinks nor swells. Now there are more glucose molecules inside the cell than outside. This cell is in a hypotonic solution. The solute molecules attract water molecules, so fewer water molecules are free to diffuse across the membrane. In a hypotonic solution, the concentration of free water molecules is higher on the outside of the cell than the inside. Osmosis occurs as water molecules move down their concentration gradient, entering the cell. As a result, the cell swells. Now let's see the effects of a hypotonic environment on animal and plant cells. Animal cells lack rigid cell walls. When they're exposed to a hypotonic environment, water rushes into the cell and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell and it will burst open or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell, which maintains osmotic equilibrium and avoids lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to a hypotonic environment, water rushes into the cell and the cell swells, but it's kept from breaking by the rigid cell wall. The pressure of the cell pushing against the cell wall makes the cell turgid, which is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Now let's look at the effects of a hypertonic environment on plant and animal cells. Since animal cells lack rigid cell walls, when they're exposed to a hypertonic environment, water rushes out of the cell and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to an isotonic or hypotonic environment, water re-enters the cell and normal functioning may be restored. Since plant cells are surrounded by rigid cell walls, when plant cells are exposed to a hypertonic environment, water rushes out of the cell and the plasma membrane pulls away from the cell wall at multiple places. This phenomenon, called plasmolysis, causes the plant to wilt and can lead to plant death. If plant cells are returned to an isotonic or hypotonic environment, water re-enters the cell and normal functioning may be restored.
So as you just saw, the movement of water from an area of higher concentration to an area of lower concentration of water is called osmosis. And so water molecules are going to equilibrate free water molecules on either side of the semi-permeable membrane when solutes can't move. And this movement of these free water molecules to equilibrate water to solute concentration is called osmosis.
Alodia is a water plant whose thin leaves are only 2 cell layers thick. This 1,000 times magnified view focuses through several adjacent cells in a leaf. In the lower cell, you can see a nucleus with a smaller nucleolus inside it. The huge central vacuole is visible in the middle cell. Chloroplasts can be seen in the stream of cytosol as it sweeps around the cells. Because this is viewed using differential interference contrast or Nomarski microscopy, the scene appears three-dimensional. Credit Michael Clayton, University of Wisconsin-Madison.
And we will look at Elodia in lab. So you'll be able to see the chloroplast moving through those when we look at them under the microscope in lab. So a review of our tonicity. So tonicity is the ability of a surrounding solution to cause cells to either gain or lose water. Hypotonic means there are fewer solutes outside the cell than inside. And so the water is pulled in to equilibrate. So it will cause the cell to burst if too much water is taken in. Isotonic means they are equal. Hypertonic means there are more solutes outside the cell than inside the cell. So it'll pull water out of the cell and cause it to crenate or shrivel. Other organisms, such as parameciums, will actually have contractile vacuoles that will allow them to osmoregulate when they're put in either hypertonic or hypotonic. So it helps them to regulate their water balance using the contractile special vacuole in them. With plants, they do not burst like our animal cells because they have that rigid cell wall. And so when plants are placed in a hypotonic solution, it causes them to become turgid. So this is turgor pressure. This is what allows plant cells to stay upright, gives them their firmness. With an isotonic solution, they can become flaccid and limp, may wilt, and when put in the hypertonic solution, they definitely lose water and will wilt and undergo something called plasmolysis, which will cause the cell membrane to pull away from the cell wall. Also with passive transport, and again, passive transport does not require energy, and it moves with its concentration gradient. So passive transport does not require energy, and it moves with its concentration gradient. Some types of cells, gases, water, nonpolar molecules can pass directly through the plasma membrane, but charged particles or larger molecules or ions, those charged particles will require a protein, a transmembrane protein. And the two main types of transmembrane proteins that are used to allow those hydrophilic or charged particles to move through the plasma membrane are channel proteins, which just form an open tunnel through the plasma membrane, or carrier proteins, and they change conformation, so they carry it across by changing their conformation. But in both cases, we are still moving with the concentration gradient from areas of higher to lower. We're just using a protein to get across the plasma membrane, so it is facilitated diffusion because it is facilitated by either a carrier protein or a channel protein. So this will end our talk on passive diffusion, and in the next talk we will pick up with active diffusion. Hope everyone has a good day. Let me know if you have any questions.